CRISPR could be responsible for a new era of genetically perfected plants, animals, and even humans, but what the heck is it? Hello there, imperfect humans. Trace here for DNews. A few years ago, something called clustered regularly interspaced short palindromic repeats, or CRISPR, burst into the scene. It worked so well, scientists began issuing ethical statements about its use. CRISPR cuts DNA strands with unprecedented accuracy and simplicity, allowing geneticists to directly edit any of Earth's organisms however they like. CRISPR could be used to engineer disease-free organisms, formulate high-yield crops, or even cure genetic and hereditary human conditions. Of course, it could theoretically also be used to let parents pick their kids' sex, eye color, height, or whatever. In the end, CRISPR is, as one Nobel scientist said to The Independent, jaw-dropping in its efficiency and simplicity. It all began in the late 80s when some Japanese scientists were looking at bacterial DNA. They spotted repeated palindromic patterns. A palindrome is a mirrored set of characters like race car or noon, though DNA only uses A, C, T, or G. In the 2000s, scientists realized these repeating characters were part of an ancient bacterial immune system. The palindromes were framing the DNA of viral invaders. This viral DNA is used by bacteria's immune systems like a most wanted poster. It would detect an attack, go look at the most wanted poster section of their DNA, figure out which virus was attacking, and then create an RNA defender to fight back. Now that the RNA knew what to attack, it needed a way to do so, which is where Cas9 comes in. Cas9, or CRISPR-associated protein 9, is an enzyme that unwinds DNA and then cuts it up. Now the trick is that cutting process works with more than just viral DNA. Once scientists figured out how this process worked, they learned how to use it to cut out and replace any DNA sequence. I know it's confusing, but think of it like our own immune system. The RNA are like antibodies, tagging the invaders, and then the Cas9 eliminates them like our white blood cells do, except instead of the cellular level, it's even smaller on the molecular level. It's tiny. After word got out, scientists everywhere began to make their own RNA targets, wrap them in Cas9, and then send them out to cut DNA, like our own private RNA army. CRISPR-Cas9 is so accurate, it can recognize as few as 20 base pairs, meaning scientists could cut single genes out of a DNA strand. After the strand was cut, it would self-repair, disabling the gene. But if scientists injected replacement DNA, that would fill the space instead. This allows us to swap in DNA wherever we like. You want corn with genes to fight bacteria? Cool, can do that. Want fish that glow? Grab some DNA from phosphorescent algae, toss it in there. This technique is so simple, it's scary. It's paving the way for widespread genetic engineering, and it's kicked off a media frenzy, like when Chinese authorities announced that they'd edited human embryos, although they'd not allowed them to grow. Craig Mello is the co-laureate of the 2006 Nobel Prize for Medicine, and he told The Independent it is a triumph of basic science, and claimed it was even more important than his discovery that had won him the Nobel Prize. But because it's so simple, scientists are calling for ethical oversight all over the place and the need to align science with public support. Now, CRISPR-Cas9 isn't perfect. Sometimes the process will target the wrong section of DNA and cut there, but a new paper in Nature describes how scientists altered the Cas9 enzyme, reducing off-target mutations to undetectable levels. So they improved on evolution, and now it is perfect, and even more scary. Thanks for watching DNews today. Do you want to know more about these Chinese scientists and how they altered human embryos? Check out the video that we did on that. Julia and Julian, our genetically perfected couple, checked it out over here.
A DNA cutting enzyme called a nuclease can be put into a cell that breaks the DNA at a specific place and takes out the faulty or unwanted gene. The DNA can use a synthetic sequence that replaces the faulty one as the strands are put back together.
What do you think about CRISPR? Does it freak you out? Is it awesome? Let us know in the comments. Keep coming back here to DNews every day. Make sure you subscribe. I'll see you next time.